Hello, Jesse.
Morning.
How are you?
What's that?
How are you?
Oh, okay. I haven't done a lot directly for DevoWorm stuff, but are we? I don't know if you submitted the abstract yet for DevoWorm stuff.
Yeah, I did for 2 Neuromatch, correct?
Yeah.
Yeah, I did. I just submitted it, I think, this weekend.
Okay. Because we can talk elsewhere, I guess, about the other one for the other group, but yeah, I haven't done a lot with DevoWorm yet, but I will be doing more with them. So.
Oh, that's okay. Yeah, I think that's it's fine. I think the Neuromatch stuff is keeping you pretty busy, and then the Neurip stuff we're doing with the other group. So it's like kind of taking all the time right now. But yeah, I think that should be something good will come out of that. Hopefully,
Yeah.
that work is a little bit maybe. I don't know if it's far afield, but it's definitely something they probably won't have another similar talk of at Neuromatch for the stuff that for DevoWorm that I'm proposing. So we're putting in.
Yeah, that looks good.
Hi, Dick.
Good morning.
How are you?
Hi. Okay, I'll do some quick rearranging.
Okay.
Yeah. One fellow wanted to meet right in the middle of your meeting.
Oh, okay.
Okay.
All right.
Oh, Susan can't make it today,
Okay.
but I hope you will schedule with her a repeat of her talk. Okay.
Oh, she just gave a talk last week.
Oh, it was last week. Gee,
Yeah.
Do you have it recorded?
I think it is recorded. I didn't know. I don't know if I sent you a link. I can send you a link again.
Oh, please do so. I get hundreds of emails per day. Some get buried.
Yeah. Yeah,
Okay.
it was a pretty good talk. It was on her optical imaging work.
Oh, okay. Okay, good.
Yeah. And yeah, I got a fairly good feedback. I mean, we don't have any other experts in the group, so it's.
Oh yeah, last week I had a bad backache that lasted a week. It took a while to go. It's like, oh,
Yeah.
I seem to be recovered now.
Oh, good. Yeah. I mean, I gave her some practice in giving the talk. It was pretty interesting to I think other people.
Okay, good.
So good. Well, welcome to the meeting. Sorry I have to keep shifting the time, but I had a prior engagement that was at one time and then it shifted to another time. So I have to make that meeting. So I have to move this one. So I hope people are. If you're not watching, or if you're not at the meeting, you're watching this later. So that being said, we're going to talk about Hacktoberfest, which is the event going on GitHub, where if you make a certain number of commits for the repository that's participating, you get like a free T-shirt from Google, and that's enough incentive for some people to make commits to things. So it's there's a whole marketing campaign around it, and we'll talk about that in a bit. Also, I have a presentation on multivariate analysis that I'll present. I don't know if anyone else is going to show up, but we'll do that at the end so that we have maybe if people show up, they won't miss anything. And then Dick, now you emailed me this week about that you had a problem, and that problem was you have a lot of ideas, but you don't know how to implement them in like software or something like that.
I know how. I'm just extremely slow.
Okay. So yeah, I mean, I guess. Oh, there's Krishna. Okay.
The, pro-
Was that?
the problem is learning a language without anybody around to help is very difficult because all computer languages have folk knowledge, which is not transmitted with the language itself.
Yeah, yeah. So I mean, yeah, that's a problem. I mean, in a lot of like, I mean, there are people who go online and chat about different problems. Usually, it's like if you're in a class, you might have a homework and you solve the
Yeah,
problem.
I remember I taught Pascal, and then I attended a class for maybe 20 years later, and I skipped after the first day. I didn't understand a word this person said. So, there's a lot of assumed knowledge.
Right. Yeah.
Okay. I mean, I used to speak Fortran 4, but it's ridiculous. It's now quite dead language.
Yeah.
And the subsequent Fortran is pretty opaque too. The one that they have now.
Oh, the newer version of it.
Yeah, there's a new version of Fortran, but for a Fortran 4 programmer, it's a lot to learn.
Yeah.
So, anyway, I tried Mathematica, and so far my code looks like Fortran 4. And the problem is when it doesn't run, it's sometimes impossible to figure out what went wrong.
Right.
And I'll be running a program and the output looks fine. I make one minor change and there's no output. What did I do wrong?
Yeah.
So, anyway, that's my dilemma right now.
Okay.
Could be fossil brain, but.
Well, I think it's very hard to get because languages move so fast.
Yeah.
Just kind of try to stay up with the state of the art, but it's always a challenge.
Oh, I know. I once had a book that had 800 computer languages outlined in it, and Pascal was not yet included. I'm sure if you made a similar book now, it'd be twice as big or more.
Yeah, definitely. So, I mean, I've done a lot of stuff like in MATLAB, and I've done a lot of stuff in Python, which is a newer language. So, I mean, that's what I'm familiar with, and of course now Python seems to be like the open source standard. So when we go to Google Summer of Code, for example, the standard is usually Python, and it's not that hard of a language to learn, but it's a bit hard to really kind of get into it. Like, how do you do X?
Yeah.
So you have to put together the pieces from like you figure out, okay, this requires a loop, or this requires some module. You have to load it in, and you have to get it running, and then so it's a challenging thing. But fortunately, there are a lot of tools out there that are available for people to make their work easier. So I think Jesse was part of Neuromatch this summer, and Neuromatch it's a summer school, and they're running it virtually, and they're trying to get people to run simulations in, I guess, Python. And so, Jesse, do you want to relay your experience on that?
I kind of missed the setup for that. I heard Neuromatch, but what was...
Well, just how you were able to handle like a lot of the notebooks, and how did you handle programming in Neuromatch?
Oh, I mean, they had a... Let me turn this off. They, I liked what they did, and the spirit of it was very good. The execution of it was a little bit weird sometimes because we basically did everything in Google Colab. Um, and I really liked they had very good videos. I really liked most of the tutorials, but it was just a little bit. There was a little bit of ping pong back and forth when you're trying to learn or write code within the collabs. That was probably the strangest part for me. But overall, like what they were trying to do, and I'm still. It's a bit delayed with everything else happening right now, but I'm still working on my own slow pod going over the material again. But I really liked that they had a very good. It's the best blend of topic overview, specific tutorial. Here's the code. Get to work with it in a Google Colab, and even work with other people. And when it's done right, it's a very good way to to engage the code and get some direct connection. It was hard with the fact that there was just so much content and it was very difficult to get to the level of depth and the amount of time. Like we had one day on reinforcement learning or like it was very extremely oversaturated in terms of material. But it's a reference you go back to and look at again. And I think it's definitely setting the bar for that kind of a learning environment in the future. I'm not sure if that's what you wanted me to comment on, but it's what came to mind first.
Yeah, I think that's a good so. Let's back up and you mentioned Google Colab, so they actually have notebooks now that you can execute code in. So there are these notebooks. I think we've talked about the Jupyter notebooks in the group previously, where you have these little kernels, which are these little windows, and you put a piece of code in and you can run it in real time. So you install this editor on your machine for Colab or for Jupyter, and it gives you this like composition notebook. And each part of the notebook, there's a kernel or a window that you can type in some code, enter some code, and run it in real time so you can test it to see if it works. And so we've used the notebooks in Google Summer of Code for a lot of things. They're very convenient. And like Jesse said, they use them in lesson plans for like scientific simulations. So in his case, they were doing like looking at like, neuroscience simulations or other things. And, it allows you to pull in, it allows you to do everything you would do in like something like Python, but it's very interactive. And so we also so Colab is one example. Jupyter is another example, and those actually run not just with Python but with other languages as well. So and then in our other group, we've for Google Summer of Code, actually we've done programming in other languages. So Python is a standard, but it's now proliferated to, I think we had one project in something called Kotlin, which is like a offshoot of Python, and then another one in Julia, which is actually a scientific simulation language, which is up and coming. So you can see that there's this proliferation of languages even now with just within Python and Python related stuff. So it's always challenging to get things like moving and get things keep on top of things. But I think the notebooks are a good way to approach the problem. I think also having like one or two standard languages within the research group is another good way to do it. So in terms of Dick's problem, I mean, we still haven't really addressed like how do you make this easier, like for a single researcher to go in and say, I want to use Python for this problem. How do I put it together? Although I think Jesse mentioned in Neuromatch they have these tutorials, so they work very hard to create tutorials on different exercises in Python. So you might have an exercise on how do you make a simulation of a cell or just a cell body. That could be done as like a tutorial. People could download a notebook, and then in the notebook it would show them the steps annotated how to do it, and they could run through it, executing the windows as they go through, and it would show, and it would be, an easy way to really kind of understand how the code works. I didn't bring any examples with me, but I did not approach this conversation. So, but I think I'll try to get some resources on that and send them to the group. So,
Hi, Bradley.
hi, Hare Krishna.
Yeah. How was the annual conference of OpenWorm?
It was oh yes, the annual. So this last week we had the annual conference on OpenWorm. It was the year. So every year they have a get together of everyone on the board and all the senior contributors, and they present on different things. And I presented on some of the stuff we've been doing in this group. We had it was very interesting because you don't get to see the board members like once a year. And there are a lot of connections there with like venture capital and other areas of like business. So they're trying to. I think Stephen Larson, who's the person who's in charge of coordinating a lot of the OpenWorm stuff, he's trying to get a board together that really is going to push the foundation forward. But so we talked about the different projects. We talked about some other business. So it was a pretty good meeting. They were really interested in what's going on in DevoWorm. So, I presented a number of the things that were going on here. And they were pretty impressed. But, overall, it was a pretty good meeting. And you get to see people, you only meet them in Slack every once in a while, and then you see them on video conference. Because OpenWorm used to have general meetings where everyone would show up, but that doesn't happen anymore. So, I wanted to get back to the conversation about the learning code and all that. So, Krishna, you've been contributing to the DevoLearn data science tutorials. Can you tell us a little bit about that?
Yeah, we generally I contribute in the field of data science. I put up tutorials and all, and I guess 2 or 3 days back I made another, you can say, folder for important links. I've added few of the pandas framework exercises, and I think I pushed an issue for making Hacktoberfest label. Did you make it, Bradley?
Which one? The issues?
Yeah, I created an issue regarding creating a label for Hacktoberfest.
Oh yeah, I've been looking over those issues. I haven't really addressed all of them yet, but I'll get into that in a bit.
So
Yeah,
that we can get Hacktoberfest contributions also.
yeah.
And I shared our our organization in my college. Now I also put a WhatsApp status so that we get more contributors.
All right.
And I have to send you the paper also. I forgot to send it.
Oh
So
yeah.
I was writing about reinforcement learning, SARSA and expected SARSA. It's taking more time than I expected it to take. Maybe all that work from home thing has affected the efficiency of the work.
Yeah, that's fine. Oh yeah.
I've become quite lazy.
Dick says Oktoberfest is so that's Oktoberfest is a German holiday, like a month long, where they have beer and everything. And Hacktoberfest is a variant of that. We'll talk about that in a minute. So yeah, I guess Krishna, you had some contributions of some data science demos. So yeah, in the DevoLearn group, then in the DevoLearn organization, we've been putting together some demos for data science. So there we have some notebooks as well that show like demos.
One more thing. It's not all about Python. I have pushed also SQL and Bash code, command line Linux and SQL also, because these are the part that people often ignore, because SQL is the basic of, you can say, there can't be data science without databases.
Yeah, yeah.
Yeah, also, and now I'll be pushing R code, and I was working on some genomic data analysis in R. Once I draft all the tutorials, I'll be pushing a 6-7 you can say lesson like structure, where it would be for beginners to how to analysis, you can say gene analysis in R language, because most of the time people only work in Python, and like things like SQL and Bash and R are often ignored. So I wanted DevoWorm to be a cocktail of all the things, not just Python focused.
Oh yeah, yeah. That's what we were just talking about. We were talking about like Dick was kind of learned Pascal at one time and did a lot of stuff in it, and then eventually the language kind of evolved away from where he was. So it's and that that just happens. I think with Python, it's happening to some extent where it's changing a lot. So you always have to keep like on top of it. And but yeah, I mean, I think that's good that we have tutorials for that, and so that's another place to go. I mean, it's
I
hard
had
to
a,
do. Yeah, it's really hard. What was that?
I have a question after you're done with that about
Oh, yeah, you can go
DevoWorm.
ahead. Yeah, ask.
So I was looking at it again. I just looked at it just now, and I see the data science demos, and there's also the education repo. And I was just wondering, is there a specific divide? Like, what's something that should be in education? What's something that like is data science demos specifically for data science related things? Like if I was going to post, I saw something. There's a resources section, and I was going to put a resource to it just now because I know I worked on something during in the past the learn neuroimaging thing, and I'm not sure if that would be more uh, learning neuroimaging, which I did with one of the brain hack things. They have the whole website and the GitHub repo, which is a really great set of resources. But would that be like would that be a data science demo resource thing, or would that be more just a general education thing resource to put in the education section?
Might be an education thing. I guess we started the repos. Let me share my screen and show that just so that we can see what I've been mentioning this a couple times now. And so here's DevoLearn, and this is data science demos, and this is education. So I think education we have nothing yet. Oh, we have the Journal of Open Source Science paper, which is still going. We're trying to get it done. I've had some things come up in the interim, but this is where we would put this. So this is like scientific education content. So this Journal of Open Source Science paper is like on the DevoLearn platform, but kind of like a paper that explains it and gives you more reference, further references for like what's going on in that. So I think probably what you're talking about would fit into this repo. And so it would be
Okay.
like, yeah, it would be because the data science demos I think are largely going to be about just like quick tutorials on different topics. Like, if I want to know how to do like manipulate some data or maybe run some Python code, this is a place. Okay, Dick. A place for you to post those. I mean, that's what I was envisioning. In these, these repositories always have some sort of like drift where they like they get too far afield and they have to reorganize it. But I think that's a good way to do it. Like if it's sort of data science, if it's like how do I manipulate data or analyze data, something specific to that, it would be that's where it would go. If it's maybe more of a tutorial on like neuroimaging, which is not really in the scope of what we are doing with data science, then it would be an education because it's useful, but it's like a little bit outside of the main area.
Yeah, yeah. And I know there's specific things within the other resource for data stuff. But okay, I don't know. Maybe I'll make a resource subsection in the general
resource
section.
yeah. I created the resource section mostly for, for example, for important GitHub repos of other people, or if, for example, like Free Code Camp is having a great video, so you can share that video in the resource section. Things that we don't own is basically for that resource section. Things that we don't own, and yeah.
So let me
We don't have take the credit of other people, so they can be put in such way like other resources, external resources.
right. So yeah, again, this is the if we go to the data science demos, we'll see that so we have networks, resources, and tutorials. So the resources are here. This is like for things that might be background reading. Networks. We want to be careful with attribution there. We don't want to. And then this is networks. This is stuff from I think that Mayok committed from some of his work on networks from this summer. There's more to put up there, but I haven't put it up there yet. Tutorials, of course, these are the ones, the straightforward code tutorials. So this one here, command line basics, IPYNB. This IPYNB is a notebook, and they don't render very well in GitHub. But if you download it and have a editor, you'll see that it's basically like it's a editor with some windows, and you can put code in and run it. This is a different format for a tutorial. So this is for this is Krishna's tutorial on SQL, which is a structured query language. And so this is actually a nice way to teach code, because you have this description of what it is, and I think it's pretty accessible. Then you say, what can it do? You have a bunch of things that it can do. Then you say, create a table. That's an example of something you can do. Then you have some, so then you have just an example of doing this. So you have this, it's a little bit vague in terms of like where you bring the data from, but it's a good way to show what it can do, just as a specific set of examples. And then inserting new values and updating values, deleting record, and putting memes in are kind of a way to soften the blow of learning a pretty dry subject. So I think and then you have references down here. So I think that's a good start for, demos. I think there's a lot more to do with demos. Dick says, my problem was that 40 years ago, I left all programming to my students. That's kind of what we've been doing with the machine learning stuff here in Devo. Like I've been getting students who are really good at machine learning and recruiting them to work on some of this stuff for DevoLearn. And I don't know if I really understand like all the machine learning. So it's like I'll probably run into the same problem there with that. But it's moving so fast though that it's just really hard to keep on top of it. But I guess that's our trying to do a lot of this stuff with tutorials and everything, so that we can make it at least somewhat accessible, to people. And even if you know code but you don't know the language that you need, having a tutorial there is nice. So I think that's good. I think we'll follow up on this discussion. Maybe we'll talk about maybe some specific problems that maybe Dick wants to solve, and maybe we can work out some. Maybe we can like post them in a way that's maybe recruit people to work on them or something like that. I don't know if that's something you'd be comfortable with, Dick.
Yeah, sure. The first two, I set 4 problems, I guess. The first two, some people responded to. We'll see if they do anything with it. But
all right. Yeah, you sent me some problems via email.
Yeah, I think in the context of this group, model for motion of diatoms might be most apropos.
All right.
I can state the problem very simply. If you watch a diatom, it moves in a jerky fashion. And if you photograph it with a high-speed camera, it still moves in a jerky fashion.
Yeah.
Okay? Suggesting that the motion itself is fractal. Okay, now, what we know about fractals in biology is at some point you get down to the molecular or atomic level, and we can't be fractal anymore.
Right.
Okay? So the problem here is to take a bottom-up approach and try to simulate the motion of molecules that we think might have something to do with the motion of the diatom, and see if that motion is jerky.
Yeah.
And then what parameters would allow us to match the model to observations.
Yeah.
Okay? So one hypothesis is that the diatom actually moves by like a rocket ship, except that it moves like a rocket ship that was proposed, about 70 years ago. And that is a rocket ship that drops atomic bombs behind it and uses the explosion of the atomic bombs to accelerate. Okay?
Yeah.
So the idea is what the diatom may be doing is setting off a whole bunch of tiny molecular explosions. And maybe if we modeled that, we could see if that works.
Yeah. Yeah, that would be good. I mean, just to, get a problem that we can easily, how would you attach code to this problem? And that seems like a standard homework problem you might encounter in a class.
Oh, okay. Yeah, I generally default to what are called lattice gas models.
Okay.
Okay, which basically involve discretization of space.
All right. Yeah.
Okay, and of events. So there may be other approaches, continuum approaches, etc. But in any case, that's the basic problem. Can we make a model that imitates the actual observations of the jerky motion of diatoms?
Yeah. Yeah. So it's sort of an approximation problem, but you have to pose it in a certain way, like a model, and then you have to put code to make it
and then let's see if the parameters that would match the observations are reasonable.
Right.
And there are other things you can do, like try to make predictions, like how would the diatom move against a force, things like that. So you could also try to do simulations of experiments that are plausible in the lab. And there's a classical experiment, for example, going back to, I think, maybe the 1960s, done by Margaret Harper. What she did is ran a diatom into a very fine fiberglass, and it bent the glass. So she could measure the force that the giant diatom could exert. Okay?
Yeah.
Now, more refined now would be, can we predict the force versus time if the motion is jerky? Okay? Yeah. I mean, things like that are ways of, in other words, trying to bring this model to a test against reality.
Yeah.
Or an alternative model. The one alternative is that the diatom trail, the gooey stuff that they leave behind, gets stretched and snaps. And as it snaps, it's like holding on to a rubber band and letting it go. Things move suddenly very fast.
Yeah.
Okay.
Right.
So this is another model, or a model might combine both and see which is more important.
Right. Yeah, that'd be great. And then, we could, then there's also the thing about, how do you evaluate across models? That would be I think something that would be like sort of beyond the immediate problem, but
Well, unless you combine the models and then just try to evaluate different parameters.
okay, yeah,
Okay, now this, we don't know how universal this is. I mean, if you look at Bacilleria, when the cells move, they look like they're moving smoothly, but nobody's looked close enough to see if the movement is actually jerky.
right,
Okay, but maybe we can get Thomas Harbich to do that.
yeah. Yeah, he generated a lot of good time series for the paper.
Yeah, but is he prepared to go to, how should I say, most cameras will run at maybe 10 frames a second. We ran a camera at 890 frames per second. And as I said, even at 890 frames per second, the motion was still jerky.
Right.
And 890 is modest. You can easily get cameras that will do 10,000 frames a second, and some will go up to 1,000,000 frames a second.
Yeah.
Okay, so, I don't know what Thomas has available, but maybe we can inspire him to get a hold of a high-speed camera.
Yeah, yeah.
I mean, he's doing some very small colonies, just a few cells. And whether or not that motion is smooth or jerky could be determined. Okay?
Yeah. Yeah.
So it's kind of fun.
Yeah. Well, yeah. Well, yeah. Let me put that together. We'll talk about that more like next week.
Yeah.
Or yeah.
The result. Sure.
I'm not from biology, so my question can be quite naive. But do this diatom collide?
Yes, of course.
So is there any change in their structure or something like that?
No, because the structure is that the cell is inside a silica shell. Okay, and silica at that scale is pretty rigid. It's like glass. Okay,
So yeah.
So you don't expect any significant change of shape when they collide. There are experiments where they collide with a beam of light and then reverse the direction, but that's a different matter.
So if you observe a single diatom particle, so does it also show jerky behavior? There can be a possibility that because of their collision, their rapid change of momentum.
No. A single diatom, the observations of jerky motion have only been made on single diatoms, which are isolated from other diatoms.
Okay,
No question. Okay? And no question mark, I should say. They are, they are isolated. They've been just single diatoms moving around on the surface.
Thank you.
Okay?
Okay.
Yeah.
That sounds good. Yeah. So thanks for the. Yeah, we'll talk more about that in the coming weeks. I think it's a good conversation to keep going and see what we can do. Now I wanted to talk a little bit about Hacktoberfest, and I know the main people involved in that aren't here today, but
probably because they got drunk.
Yeah. So we had so there's this thing called Hacktoberfest. Let's see, it's right here. I have the image. Oh, let me share my screen. I didn't do that yet. So there's this event called Hacktoberfest. Like I said, it's through the month of October, and GitHub hosts it. It's a play on Oktoberfest, of course, which is a German holiday where they celebrate the entire month. And so this is the Hacktoberfest. This is for the DevoLearn, or, for all of DevoWorm, but we're focusing on DevoLearn for this event. So the idea is that throughout the month of October, people can go to the GitHub repository and make commits to the repo. It could be anything that they want to do. Add in some text, add in a tutorial, make changes to some code, and then if they get like 5 or more commits, they get something free from GitHub. So it's a T-shirt usually, and it has the Hacktoberfest name, and it has like GitHub on it, and it motivates people, I guess, to do some commits. So we have in let's see. So this is DevoWorm. This is where the action is occurring for the Hacktoberfest, and it started the 1st, and we've already had I think about 12 people contribute to. And it is mainly in DevoWorm here in this repo. So they were. I think this was driven by Mayuk and Ojwal, where they, I think, talked to people they knew at their school, and they started to make commits to the repo. So we have a bunch of commits that are, I don't know if you can see it under actions. No. Pull requests. We've had like probably about 12 or maybe 10 pull requests in the last couple days. So it started on the 1st, and it's going to continue through the month. And we've already had a pretty good amount of interest in it. So one of the things you can do if you want to commit or if you want to participate in Hacktoberfest, you can go to the issues in any of the DevoWorm repositories. In this case, we have DevoWorm has a good number of issues to look at. And you look at these issues that have been generated. I think Mayuk is mostly generating these in the DevoWorm part of DevoWorm. But there are other. I'm not sure if Ojwal's done the same for C. elegans DevoWorm. There's one issue open here. This is add contact info. So this is something that I don't know if it's a question, but basically, people will go through these issues. They'll pick an issue, and then they'll address it. And what you do is you make a fork of this repository, and then which is basically just your own version. You would go to fork, and it would create your own version of that thing on your GitHub account, and then you would make the changes, and then you would submit them back to the main repository. And so that's called a pull request. So you're taking a copy of what is on there right now, you're pulling it down to your own account, you're making the changes that you want, and then you're sending it back for approval to the main account. And then if it's good, and a lot of these are pretty good, these contributions, then we merge them. And so here's where the pull requests come in. People will make them. They'll appear here, and then we can review them. I've been involved. Ojwal and Mayuk have been involved in this, where we're all reviewing these changes, and basically just to make sure that they're not malicious or that they're not junk, which can happen. But and then a lot of times there will be like errors that they make or things they need to add that you can suggest they make before their pull request is accepted. And then that happened, I think, twice in this batch of pull requests. So we have a lot of things, that it's a nice process for sort of managing version control. So, we have files that we are constantly making version changes to, and GitHub is designed so that you can make those changes without overwriting things and being able to track all the changes. And it's very effective. That's why we do a lot of things on GitHub. So that's how basically how a pull request works. If you want to contribute to this, you're welcome to do so. This is just something that Google puts on GitHub puts on as a way to encourage participation in open source. So hopefully we get a lot more of these. There's also, I think, an issue we had to solve about verifying the repo for participation in Hacktoberfest, but I think that's been solved. So now I think if people contribute 5 pull requests during the month, they get a T-shirt. But I'm going to try to recognize people, and this is our list here. I'm making a list of contributors who have made pull requests. So we have a bunch of user IDs here, and these are all people who have issued pull requests. And these are the status of the pull requests. So they've all been accepted. A couple of people have made 2 pull requests already, and they're just like little changes, I think. But and then I'm making a list of further interactions. So Krishna's on here. He made a pull request. I think it was before our Hacktoberfest, but I put him on the list, and I put Malvi M on the list because she contributed something to the tutorials. And so I'm just making a list of people who are contributing to DevoLearn and some of the other repositories in DevoLearn, and following up to see what they're doing. So Malvi M has joined our Slack channel, and Krishna's, of course, joined the lab meetings. And so I just want to see how this is pulling people into the group and where they're going, if they're sticking with it, or if they're, just kind of casually making a pull request or whatever. And so that's a good census tool. And I think, it'll drive interest in the group as a whole. So, hopefully some of these people will join Slack, or I don't know if maybe we can have some discussion on GitHub about things. If that's where they're living, if that's where they're checking in, maybe we can get more people involved in, some of these problems that we're posing in the meetings. But it's a good thing to know, and I think it's a good thing to get people involved in this. So that's all I have to say about Hacktoberfest. This is the intro page in our DevoWorm GitHub. So if you want to see the basic description of what it is, here it is. Again, it's just simply this, you check the issues, you go and you try to solve the issues, and you get connected to the community. If you don't really have faculty with GitHub, and some people don't, especially if they're coming from the biological side, because it is more of a computer science centric tool, then you can contact people, or join the Slack channel, and that's another way into the community. I don't want to exclude people who aren't really, that don't have much to say in terms of GitHub commits, because that is, again, maybe very code centric. So if we have people who want to commit in other ways, it wouldn't really be part of the Hacktoberfest system, but we'd still be interested in hearing what you have to say. And so that's a good overview, I think, of that. Are there any questions about Hacktoberfest or any of the contributions?
I had a general question. I saw that I just fully signed up for Hacktoberfest right now, and they do offer these things that are events, and I wasn't sure if that's something that was in the works for DevoLearn or other orthogonal stuff. I'm not sure what an event entails, or if it's just a presentation exactly, but
I'm not sure either. I think some of the organizations might have events surrounding a Hacktoberfest, maybe their get-togethers or something. It's just a way to encourage people to participate. So they might have things where they're having meetings of people, maybe open events where people can come and interact at a little bit higher level than GitHub commits. I'm not really sure. You'd have to show me an example, though.
Yeah, it looks basically it looks similar to an Eventbrite page from what I'm looking at.
Oh.
You can kind of make under the DigitalOcean banner, you get to sort of make a Eventbrite, kind of organize meetings around a specific project, I guess. So
Right.
I don't know if that was something that other people in DevoWorm were trying to do or not. Or it's not something to talk about now, but I might mention it later.
Well, maybe like later in the month we could certainly try something if we wanted to do an event.
Obviously right now it's
Yeah. Well, yeah, I mean, it's like I don't like other like some organizations do it every year and they're like prepared for it. And so I don't, but we'll see if there's some interest in it. We might send out like I might send out a message to people who've contributed and say, "Would you like to come in?" We might have a meeting where we discuss their contributions and then maybe what things that they might follow up on, and it might be things that are more like more involved projects or things that aren't just like isolated commits, so that maybe they can get a little bit more exposure to DevoLearn or DevoWorm, you know. So that's, I mean, that would be good, but we'll talk about that later. So last thing I wanted to talk about today is this presentation that I wanted to give. And if you have to leave at 10, or at the top of the hour, that's fine. But I just wanted to go through this. So this is something I've been trying to wrap my head around for a while now. And this is how to understand multidimensional analysis in developmental biology. So what I have here are three different methods. One is, I think everyone's heard of as PCA, or principal component analysis. And this is where you're taking a bunch of data and you're trying to find the axes of variance. But then there are two other methods that have been more recently applied to mainly molecular data. And these are tSNE and UMAP. And these are probably things you haven't heard of so much. But if you read any sort of like molecular biology paper in development or in other areas of biology, you'll encounter these methods. And so I wanted to demystify some of them. So again, I'll make these slides available afterwards, and it'll be recorded. So if you can't make the whole talk, you can go back and check it out. So principal component analysis is an exploratory data analysis technique, and people have used it for many years, many decades. It's a tool that's been developed around like just regular data. It could be any type of data. It could be like, cell measurements. It could be any type of thing. It could be social science data. And what principal components analysis basically does is it takes all of your data points and it tries to take the matrix and find a series of directional vectors. And these vectors are, the vectors that come out of this are vectors that best describe the data while being orthogonal to all other vectors. So what does that mean? That's kind of a weird way to put it, but basically what it does is it takes a bunch of data, maybe in a bivariate relationship, then centers the points, it computes a covariance matrix, and it calculates eigenvectors and eigenvalues from those. So it does this transformation into eigenspace. Then there's this where you're picking the eigenvectors with the highest eigenvalues. Then you're projecting these data, or projecting the data points to these eigenvectors. So you're taking the data, you're extracting information about their sort of core variance, and then you're taking the that model of the variance, and you're mapping the data points back to them, and then you're getting a map of those data and how they're distributed. So this is from a Hacker Noon tutorial, but there are books on this. I just wanted to give a couple of blog posts on this that maybe are more accessible. So this is an example of what people usually do. They'll take their data, they'll analyze it using PCA, and then they'll get maybe the top two dimensions or top two principal components, and they'll plot them in a bivariate graph, and then it'll show these sort of the scatter of the data, but also these groups. And the clusters are, basically they are organized along some axis of variance. They're not really like a clustering algorithm. They don't exactly match up in that way, but they give you some indication of what the variance looks like. So this is an example here of like taking a picture of a flower or morphological data from a flower and creating a PCA biplot, which are these top two axes of variance. And you can define different features in the data by looking at the different vectors. So this is sepal width, this vector. This is sepal length. This is petal width, and this is petal length. And you can define these clusters, and then you can define it by species. And the PCA analysis gives you an idea. It's sort of, looking at the shared variance and the variance between groups. So you have these species basically fall in separate clusters. So this Setosa species is very different from the versicolor species, and the Virginica is overlapping with the versicolor species. And if you were to just kind of idea it in nature, you may or may not see that distinction, but this puts some numbers on it. So again, this is how PCA works. This is a Nature Methods paper on the method. So this actually goes through a bunch of data sets that they've used to look at. This one is just kind of a tutorial of PCA and how it works. So this is PCA geometrically projects data onto a lower dimensional space. So you take all of the dimensionality of your data set and you map it to a series of dimensions that are defined by the vectors that I mentioned before. And they try to make them as orthogonal as possible so that you're getting some sort of independent set of variables, basically, that you can compare to one another. So that's the first step. This is, of course, where you can help to identify clusters in the data. If you compare it with a hierarchical linkage analysis or clustering analysis, you see that this is a hierarchical linkage method. So it forms these, basically a tree with nested sets in it. And so you can see that this matches up with this cluster analysis, but it's not an absolute map, one-to-one map. The PCA analysis will reveal features that the cluster analysis doesn't, and vice versa. So it's not exactly like a clustering analysis type of clustering. And then you have this, there's some limitations on PCA. One of them is that it may miss nonlinear patterns of data. Another is that it misses non-orthogonal patterns of data. So if the data is well organized and highly structured, it will pull out these clusters. But if it's not, then if there's there's interactions in the data or there's unexplained variance, it's going to be harder to find a good, interpretable set of images here. And that's interesting that we talk about this interpretable set of images because we don't think of it that way. We think of it as this hardcore quantitative method. And that brings us, of course, to tSNE, which is T-distributed stochastic neighbor embedding. And that's a lot of words for something that is really just like a dimensionality reduction technique. So, like I said, you're interpreting these sets of points. And you'll see with tSNE that that's actually more true, even though this is a more advanced method than PCA. So this started off, Jeff Hinton and company actually published a paper on this in 2008, visualizing data using tSNE. And this is a machine learning sort of approach. And so in that sense, it's more advanced than PCA. But it does resemble like more simple dimensionality reduction techniques. There's a technique called multidimensional scaling, which it's also closely related to. And that's actually a very simple method, but this is, this was the state of the art maybe 10 years ago. So for this tSNE algorithm, there are two steps. The first is to construct a probability distribution for high dimensional objects. So you take what PCA does and you look at the high dimensionality of your data, but you construct a probability distribution for it. And in this probability distribution, your objects that are similar in some way have a higher probability of occurring, and dissimilar objects have a lower probability. So then you construct this probability distribution. You do a separate probability distribution for objects on a low dimensional map. So you have the real data and you're trying to construct a probability distribution on that. But then you also have this low dimensional map that you want to transform these data or map these data to, that you're also constructing a probability distribution on. This probability distribution for the low dimensional map is based on minimizing what they call the KL divergence for each point. And so it's basically KL divergence is a technique that is used. It's the Kullback-Leibler divergence. This is the longhand, and it basically computes distances or divergence between two separate methods or two separate trajectories. And so you're just basically trying to find the high dimensional, like you're trying to approximate the high dimensional data, map it to a lower dimensional map, but that lower dimensional map has its own distribution, and you're just trying to match the two distributions, and you're trying to minimize the divergence between, say, a point in the high dimensional probability distribution and the low dimensional probability distribution, so that the map is as close as it possibly can be. So this produces clusters like it does with PCA, and it's a similar method in terms of mapping things back down to a lower set of distributions. So it does produce clusters, but the cluster validity is a little tricky. It requires a lot of visualization and interpretation. So KL minimization is done using something called gradient descent, which is an optimization method that is very common in machine learning, but that still doesn't solve a lot of your problems of validating the clusters. So this is like the MNIST data set using tSNE. And so MNIST is this data set that they use in machine learning that has handwritten numbers from 0 to 9. And it's like different handwriting, different people doing different sort of swirls and things with their handwriting. So the 4s look very different across the different samples. And the idea is to be able to identify all of these as a 4 in this row of 4s. So all the variation in the 4s should be, the machine should be invariant to that variation, and it should always identify a 4 correctly. Now, if you plug these data into tSNE, you actually find that you can take like data that you predict as being a 3 and put it into a cluster that's defined as a cluster for 3. So it actually does a pretty good job on this MNIST data set. It classifies those instances correctly. So all these light blue instances are 3s that are then put into the category of 3 in this map. And the 3s cluster together. Now you can see there are a couple of reds here, which are 1, I think. Oh no, 8. So you see that 8 is being misclassified as 5s and as 3s a little bit. So it's a little messy, but the idea is that the proper classification should be in one of these clusters. And you can see it looks very much like some sort of postmodern art thing where they've just kind of like a Jackson Pollock almost. And this is the problem with interpreting this. It creates a very pretty image, but it's hard to interpret. And so the basic rule of tSNE is that points within clusters are meant to be similar in some way, even if they're misclassified. But it's hard to say that if these clusters are less similar than these clusters. So these two clusters, the one at the top here and the one over here, it's hard to say whether these are less similar than this cluster. So basically, if you're looking within a cluster, it's easy to say that they're similar. And that's an artifact of the approach that they use for the KL divergence approach. But between clusters, it's harder to make that distinction. And it's an artifact of the search algorithm in the way they implement that. So there's another study that uses tSNE where they took a bunch of data sets of brain tissue or embryo and brain tissue, a lot of things from development here, and they use different molecular sequencing techniques, next-gen sequencing techniques. That's not really as important as the differences between the data sets. So this is a meta-analysis of all these data sets, and what they're going to do is they're going to map it to a tSNE model and see if they can identify different cell types. So they have these. First of all, they tried all these different methods on the data, and they got some performance statistics on this one data set, Tasek et al. So I think Tasek et al. Is what we're going to focus on here. This is adult mouse cortex using SmartSeq2. So you're looking for a bunch of classes here, and you have this thing, perplexity and random initialization. So perplexity, I don't remember what they're actually calling it, is the main parameter that you're looking for in tSNE. And it's a method of, I think it might be similar to convolution. I'm not really sure what it is, but that's the parameter that's the key. And it's a way of algorithmically sorting the data out and trying to find a good mapping between high and low dimensional space. As you can see, tSNE produces a lot of clusters here. Whether the clusters mean anything is a different issue. And so one of the things we can do is we can look at out-of-sample mapping. So in this example, we have aligned two data sets. We have a reference tSNE atlas and a data set of interneurons. And so we're able to map those interneurons onto this reference atlas. And we're able to do it fairly successfully. And so there are two, actually, tSNE has a number of parameters. So perplexity is the measure of global structure. So I remember I told you that these individual clusters are sort of, you have a very good measure of sort of similarity within clusters, but then between clusters, we really don't know what's going on. Well, perplexity, if we set this at a different level, we can refine this global structure. So it's like a top-down way of saying, we want to look at like how many clusters we have or how similar the clusters will be. But it's not really a fine level of control. It's sort of this measure of it's a general measure of global structure. As it turns out, tSNE is not very sensitive in this respect. And there's another method, UMAP, which is much more sensitive, and we'll talk about that next. Perplexity is the main free parameter in tSNE, so this is the one parameter you can play with to improve your results. But there are others, such as learning rate and number of iterations, which you can also play with to improve your result. So this was like 10 years ago. Now, in the last couple of years, we've come up with UMAP, Uniform Manifold Approximation and Projection for Dimensionality Reduction. That's a long set of words, but basically it's an improvement on tSNE. It works in a similar way, but it's better, apparently. And so they're using this more and more now. And you'll see it in papers. And I'll show you at the end that there's an atlas that uses it that involves C. elegans developmental data. This paper, McInnes, Healy, and Melville, is sort of lays out the method. So this is the sort of the seed paper for the entire enterprise. And then this is a blog post on why is it exactly better than tSNE, which explains kind of the pros and cons of the two methods. So this pretty picture here is what you get with UMAP. And if you compare UMAP and tSNE, we know that tSNE does not scale well, especially for stuff like single cell sequencing analysis. So snRNA-seq or other methods, it really doesn't do a very good job of scaling well from small to large data sets. And your average next-gen data set is very large. So we want to have a method that's robust to that size of data. Also, tSNE, we also know that does not preserve global structure. So it doesn't preserve global structure only within the clusters. So, if we look within clusters, tSNE does very well. Between clusters, it doesn't really tell you a lot. tSNE performs non-parametric mappings between higher and lower dimensions and does not rely on features. And so PCA has something called loadings, which are features that you can use, but tSNE doesn't have that. And so to avoid sparsity, which are fragmented clusters, they call them manifolds, but the manifolds just have like unconnected clusters. That's what they call sparsity. So if you have a bunch of clusters that are just very far apart, that's also bad because it's sparse. It doesn't give you a lot of information about it tells you that they're clusters, but it doesn't tell you kind of how they're related. So to avoid sparsity for high dimensional data, that are first handled by an autoencoder or PCA. So UMAP is actually bootstrapped by some sort of pre-processing technique. So as tSNE only preserves local structure, it resembles the cost function for a multidimensional scaling, which is a much simpler method. There's this lack of global distance preservation in tSNE. And so to solve this, UMAP actually uses a stochastic gradient descent model. So they use this model of gradient descent, but they use it in a way that's very different from what tSNE is using, which is they're using it sort of as a way to optimize like KL divergence and other things. So, but stochastic gradient descent, I guess, is probably better, and they kind of go into it in the article as to why it's better. UMAP also uses an exponential probability density function for high dimensional data. So, if we use like a normal distribution, we may miss a lot of the outliers or a lot of the things that are out of distribution. And if you use an exponential probability distribution in the high dimensional case, you pick up a lot of that variation that you're going to miss just by looking at, assuming that you're just trying to fit them into normally distributed categories. Because as we know, high dimensional data, there's a lot of potential for interactions. So the distance metric that's used in UMAP varies across the manifold or space, and the nearest neighbors graphs result from fuzzy simplical sets, which is a form of topological data analysis, which we've talked about in this group before, but is an interesting sort of wrinkle to this because then we're bringing in a new method, and it's a different and exciting method that might solve some of the problems that we've had with tSNE. UMAP also uses a number of nearest neighbors instead of a perplexity measure. So this is similar to information theory, where perplexity is similar to an information theoretic measure. But UMAP uses the number of nearest neighbors as a proxy or as a high-level parameter. And so the parameter dmin demonstrates a set of uniformly connected points. This is sort of this nearest neighbors approach. Using a nearest neighbors approach instead of something like perplexity leads to tightly packed clusters. So you get this more dense local information. And then they use binary cross entropy as a cost function instead of KL divergence. And I think there are some problems with KL divergence in terms of its sort of how it's sort of its rigor and completeness for what you're looking at here. So binary cross entropy is a better choice. Finally, there's this tool that is it's an interactive atlas of single cell sequencing data for C. elegans neurons in development. And so, I think it is just for development. So this is a lineage resolved molecular atlas of C. elegans embryogenesis at single cell resolution. And this is a science paper that came out last year. This is the VizCello tool. You can see it on GitHub, and there's also a Shiny app that allows you to actually just generate these plots directly. So if you go to the Shiny app, you input your parameters, and it produces these clusters, which look like sort of the tSNE clusters, but they're much denser, and there's more global structure here. So what you have is you have a bunch of cell types, and you have they're color coded, so you can see that they cluster within their groups. And basically this tells you something about how different they are from one another. So the ciliated amphibian neuron, for example, fits into a couple of clusters, and it tells you that maybe something in relation to some other cell types. And this is based on all of the single cell data that they have for the cell. So they have a bunch of sequencing data for the cell. It defines the cell's state, and this is sort of a summary of that in a low dimensional space. And so we have this two-dimensional space where we have these dots for the ciliated amphib neurons and comparing them to other types of neurons, ciliated non-amphib neurons, for example. And these gray dots are just kind of like undefined identities because we don't have all the cell types in this plot. So it actually does allow you to compare with many other types of cells. And so, for example, developmental cells born at 250 minutes seem to contribute to all future types of cell types and tissues. So we have this, if we think about it in terms of a lineage tree, the developmental cells that are born at 250 minutes are responsible for all this variation. You get all sorts of neurons and glia that come out of this. You get so that's just sort of to set this up. You get these axes like you do in PCA for this analysis, V1 and V2. these two axes represent the dimensions of a two-dimensional projection of the source data. So this is just a two-dimensional map of all this variation. And distance on the manifolds are defined by single cell transcriptional profiling of all cells. So these distances between clusters, for example, is a product of this mass profiling of a single cell and basically a summary of that distance. And so the question then is, with any of these methods, can you accurately capture that? And so people say with UMAP, it's a pretty good chance that you can capture it. But there are probably new methods that can be developed that would make this even easier. So again, I apologize for going. I see Dick had to leave, but that's okay. So I mean, I've been trying to figure out how to present that for a while, and I just wanted to give you a primer. And like I said, if you have, this is something that you'll probably encounter if you read developmental biology papers with like molecular data in them. Don't be scared off by the figures and the techniques. This, I think, is a nice primer for that. But if we have other questions about it, we can follow up on them. So any questions?
No questions, but I appreciate that overview of things because I've heard about some of those things I wasn't really sure, and some of it obviously, has come up in stuff I've been exposed to before, but I appreciate the discussion of it.
Yeah. No questions, and thanks for sharing.
Yeah, no problem. I'll share the slides later. And then, yeah. So thanks for attending. Happy Hacktoberfest. And if we'll talk on, offline on Slack. And next week we'll have another meeting. Maybe we'll follow up on the programming issues, like, how to solve a problem. And hopefully we'll have more Hacktoberfest activity.
Yeah.
All right. So, Krishna, I look forward to seeing your paper. And
yeah, I'll just send you. I have to send you one more thing also. I'll send you right now.
Okay. Sounds good. Okay. Have a good week, everyone. Take care.
Bye. Bye, Bye, Bradley. Bye.
Bye.